Hi. Just in case you didn't see the message in the chat box, I'll start the session at 6:10 so that if there are others to join, they can join before I start. So thank you so much for waiting. Good evening. Okay, I don't think there are others to join, so maybe I think I'll start now itself. So I'll just explain how this works to you in case you're joining for the first time. So this is the problem solving session for the NPTEL course, Post Harvest Operations and Processing of Fruit, Vegetables, Spices and Plantation Crop Products. So how this will work is, in this session, I'll be taking about 19 to 20 questions, which will be from this module. And if you have attended the module, if you've watched the videos, you will be able to follow along, right? So we'll take one question and then we'll try to answer it. If you know the answer, good enough. Anyways, I'll be discussing the answer also. So just so that we know, or you know at least, where you stand in terms of what knowledge you've gained in that session itself, right? So I'll be taking the questions, and if you're uncomfortable unmuting yourself, you can just write any doubts that you have in the chat box. I'll be reading them, and I'll explain whatever I know to you. And when I'm asking the question, try to guess the answer. If you know the answer, that's very good. If you don't, even then, just try guessing since it's all multiple choice questions, right? So just try guessing it, and then we'll afterwards I'll explain the answer also in case it's something difficult. These questions will be similar to the ones you have in your assignment, but not the same. So it will just give you an idea of what sort of questions will come and how you might want to answer them. All right. So just if you have any doubts, you can unmute yourself and ask me, or you can write it in the chat box. So thank you so much for joining, and I'll just get started now. All right. So let's move to the first question, which is which of the following statements about aerobic fermentation are correct? And there are four options given to you. The first option is it requires the presence of oxygen. Option 2 is it produces alcohol as a byproduct. Option 3 is it is carried out by strict anaerobes. And option 4 is it generates more ATP compared to anaerobic fermentation. 1. Alright. So let's see what the answer is then. So the answer is 1 and 4. So the question is which of the following statements about aerobic fermentation are correct? So you know there are two types of fermentation, aerobic and anaerobic, right? So aerobic fermentation requires the presence of oxygen. That is correct. Okay. It does not produce alcohol as a byproduct. That is in anaerobic fermentation. And it is carried out by strict anaerobes is also not correct because it is carried out by aerobes, right? Or facultative anaerobes also. So if you have, you can have strict or facultative. So strict means they can only use anaerobic or aerobic fermentation, and facultative means depending on the environment that they are in, they can do both of them, right? So it is carried out by strict anaerobes, that is not correct. And option 4 is it generates more ATP compared to anaerobic fermentation, which is correct. So because the lactic acid is it sorry, the pyruvic acid formed in glycolysis, when there is presence of oxygen, it goes into the TCA cycle and it produces around, I think, if I'm not wrong, 36 ATP. And but in anaerobic fermentation, that doesn't happen. So the pyruvic acid is converted into either lactic acid or alcohol, right? And that gives only a little amount of ATP. So aerobic fermentation gives you the maximum amount of ATP. Is that clear? Do you have any question in this doubt in this question? Right. So let's move on to question number 2. Question number 2 is match the type of fermentation given in column 1 with its description in column 2. So in column 1 there is lactic acid, alcoholic, malolactic and acetic acid, and in column 2 you have produces alcohol and CO2, converts malic acid to lactic acid, produces lactic acid from carbohydrates and converts ethanol to acetic acid. So which options do you think is correct? Alright. So maybe I'm not seeing an answer. So maybe we'll go to the correct answer, what it is. Alright. So the correct answer in this is option A. So that means lactic acid you can connect to produces lactic acid from carbohydrates, right? Alcoholic produces alcohol and CO2, which is B is 1. Malolactic acid converts malic acid to lactic acid, and acetic acid converts ethanol to acetic acid, right? So different organisms produce different kind of respiration which can lead to these products being formed. Okay, so the correct answer is answer E. Do you have a doubt in this question? All right, so let's move on to question number 3, which is again a match the following type of question. We have components of a fermenter in column 1 and its function in column 2. So in column 1 you have impeller, aerator, pH probe and sampling point, and column 2 you have measures and monitors pH, introduces sterile oxygen to the media, mixes the medium evenly, and obtain sample during the process. Do you want to try guessing what the answer is? Or do you know what the answer is? A. Alright, yes. So the correct answer is in fact A. So impeller will mix the medium evenly, because if you don't have mixing, what happens is there might be different kind of components in the bioreactor, right? And they might settle at the bottom. And when that happens, the cells, imagine this is the bioreactor, and the soluble compounds are settled because you have to keep it for a long time, right? But the cells are here also, and they don't get the nutrients, so you have to keep mixing it so that there's a uniform condition, and that also helps spread the gases inside, right? So aerator introduces sterile oxygen to the media. So the reason it has to be sterile is if it is not sterile, you might be introducing some other microorganism into the bioreactor, and whatever product you wanted to get, you might not get, right? The pH probe will measure and monitor the pH, because organisms have a certain pH in which they work in the optimum conditions, right? So if that pH changes, so some organisms, like in the case of lactic acid producing organisms, they keep producing lactic acid, right? And maybe your product, the product that you want, is not lactic acid. So at one point, after the lactic acid increases a lot, these organisms will stop working. Their metabolism will stop working. So to prevent that, you will have to check the pH and to bring back the pH to the optimal pH, right? And sampling point, to obtain samples during the process, so that you can measure things like how much of the medium has been used up, what is the waste products, how many cells are there, are they dead, and how much product have they produced, something like that. All right. Any doubts in this question? All right. So let's move on to question number 4. Question number 4 is, there is an assertion and a reason. So the assertion is, solid state fermentation is used for cultivating microbes on solid substrate, and reason R is that solid substrate fermentation requires a medium with above 95% water content, right? So what do you think the answer is? Either both A and R are true, or both A and R are not true. A can be true and R can be false, or A can be false and R can be true. Do you want to try guessing the answer? B. All right. Yes. So that is the correct answer. Both A and R are true, but R is not the correct explanation for A. So yes, solid state fermentation is used for cultivating microbes on solid surface. And yes, solid state fermentation requires a medium above 95% water content, but R is not the reason for A, right? So let's move on to question number 5 if you don't have any doubt in this. Alright, so the question is, which of the following statements about batch fermentation is or are correct? It involves a sequence of operation from inoculum development to production. The fermentation time can vary from hour to week. Growth rate is controlled throughout the process, and the product is harvested at the end of fermentation process. So anytime that you have any of these is or are correct questions, just keep in mind that you can have multiple correct answers, right? So one of it can be correct, two can be correct, three can be correct, and all four can be correct. So just keep that in mind, that this is an MSQ, not a MCQ, right? Do you want to try guessing what the answer is? Which of these are correct? 1, 2, and 4. Yes. So let's check the answer. And that is correct. It is 1, 2, and 4. It does involve the sequence of operation from inoculum development. That is what cells you're going to put, or what biological material you're going to put in the bioreactor, to the production. So this is correct. The fermentation time can vary from hours to weeks. Yes. um, the product is harvested at the end of the fermentation process. That is also correct, but growth rate is not controlled throughout the process, because the specialty of batch fermentation is after you add the media and the inoculum, you don't continue adding or monitoring the media, etc., right? So growth rate is not controlled throughout the process. So let's move on to question number 6, which is a match the following question. So you have to match the fermented plant food and beverages in column 1 with their primary ingredient in column 2. So you have kimchi, wine, nata de coco, and mead. Do you want to try answering this? B. All right. So let's see. Yes, B is the correct answer. So kimchi is a Korean dish, slightly like our achar, but made from cabbage. Wine is made from grapes. Nata de coco is fermented coconut water, and mead is made from honey. So yes, that is correct. So let's move on to question number 7. Again, a match the following kind of question. So you have the type of bioreactor with its description, right? So column 1 has packed bed, a fluidized bed, photobioreactor, and membrane. And column 2 has uses light energy for microorganism growth, contains immobilized biocatalyst in a tubular reactor, combines characteristics of stir tank and packed bed. And option 4 is uses membrane for suspended biomass and solid removal. What do you think the answer can be? A. Yes. So you are again correct. It is A. So packed bed contains immobilized biocatalyst in a tubular reactor. B is 3. Fluidized bed combines characteristics of stir tank and packed bed. Photobioreactor uses light energy for microorganism growth in case of if you want to grow algae, things that grow in sunlight, right? Like plants and algae. And membrane is 4, uses membrane for suspended biomass and solid removal. So you are correct. So let's move on to question number 8. So this is a numerical type question. You have to calculate the molecular flux of glucose across the membrane, right? If the molecular, so imagine this is the membrane, you have to calculate the molar flux of glucose across the membrane, that is the movement of the glucose. If the molecular diffusivity of glucose in water is 6.7 into 10 to the power minus 10 meter square per second, and the concentration gradient is 0.5 mole per meter 4 to the power 4, and the distance of the diffusion is 0.02 meter. Yes. So maybe let's try to solve this together. So what they're telling you is the molecular diffusivity and the concentration gradient and the distance of diffusion. So how much distance the glucose has to travel from across the membrane, right? So this is how you get the answer. So the molar flux, that is JA, right, is equal to the negative of DAB, which is the diffusivity of glucose in water, which is minus 6.7 into, sorry, so the minus is there, 6.7 into 10 to the power minus 10, right? And this is multiplied by this constant, which is the concentration gradient, right? So which is given in the question 0.5. The distance of the diffusion is not used in this answer at all. So that is just a trick. So all you needed was the diffusivity of glucose and the concentration gradient. And you get an answer of 3.35 into 10 to the power minus 10 moles per meter square per second. So the correct answer is option B. Alright. Do you have a doubt in this question? Alright. So let's move on to question number 9. So question number 9 is again match the following kind of question. You have to match the type of wine with its characteristic in column 2. So you have table wine, dessert wine, fortified wine and sparkling wine. And you have contains considerable amount of CO2, low alcohol content and little or no sugar. Fortified sweet wine contains added alcohol or distillate of wine. So you have to connect which wine and its characteristic to each other. So try guessing what the answer might be, or if you know the answer, just let me know. So just try seeing what wine and what characteristic it is, and try guessing from the many options given to you, the four options given to you, which might be the correct sequence. Anyone want to try guessing the answer? All right, let's see. So the correct answer for this is A. So let's see how that works out. So table wine has low alcohol content and little or no sugar. Dessert wine is fortified sweet wine, right? Fortified wine contains added alcohol or distillate of wine. So what happens is there is a limit to how much alcohol can be produced. So if you have a mashup of grapes, right, and you put yeast in it, there is a maximum amount of alcohol that can be produced by these yeast in this particular setup. I think it's about 12%. Right, I'm not sure, I need to check that. But after that, what happens is alcohol, as you know, is a disinfectant antibacterial, right? So it will start killing, if at any higher concentration, it will start killing the yeast. So the feedback loop makes it so that the yeast stop producing alcohol, right? Because they will die. So what you have to do, if you want wine with a higher alcohol content, that is fortified wine, you have to add alcohol or the distillate of wine, right? And sparkling wine contains amount of considerable amount of CO2, so that you get something like soda, but wine, right? Any doubts in this question? All right, all right. So I'm not seeing any doubts. For those who have joined now, you can just write the doubts if you have any in the chat, and I'll read them. And anything that you want to answer also when I ask, you can also write that in the chat. So let's move on to the next question. So the next question is again I match the following. Wait. So you have to match the wine in column 1 with its preparation method this time in column 2. So you have red wine, white wine, pink wine, and sparkling wine. And in column 2, you have secondary fermentation in the bottle, short exposure of grape skins to juice, fermentation without grape skins, and fermentation with skin and juice together. So try guessing what the answer might be. D. All right. Um, Anyone else has another answer? Or do you think it's some other option? Alright, I'm not seeing anything. So let's see what the answer is. And the answer is in fact D. So A, that is red wine, is fermented with the skin and juice together of the red grapes or the purple grapes that you have, right? B is 3, that is white wine, where fermentation is without the grape skin, and because the grape skin does not leach color into the wine, it remains that light yellowish color. Pink wine has a short exposure to the skin of grapes, right? So it, instead of having that dark red color, you have a softer red or pink color. And sparkling wine has secondary fermentation in the bottle. So it becomes that CO2 infused wine, right? Any doubts in this question? All right. So let's move on to question number 11, which is again match the following type question, where you have in column 1 the type of wine and in column 2 its characteristic alcohol content. So you have table wine, fortified wine, sparkling wine, and dessert wine. So try guessing the answer. A. Alright, so let's see what the answer is. So the answer is in fact A. So table wine has the least amount of alcohol, which is 5 to 14%, right? Fortified wine has a larger amount of alcohol. That's why it's 19 to 21% because you've added alcohol into it, right? Sparkling wine can, uh, yeah, sparkling wine has around 10 to 12%, which is the normal range. And dessert wine can vary, often similar to fortified wine. It's just sweeter in taste because of the added sweetness, right? So you were correct. So let's move on to question number 12 if you don't have any doubts, right? So question number 12 is which of the following statements about alcoholic fermentation is or are correct? Again, there might be more than one correct answer. So keep that in mind, right? So there are four options given to you. It is carried out by yeast such as Saccharomyces cerevisiae or baker's yeast, commonly known as bakery yeast. Two is alcoholic fermentation produces ethanol and carbon dioxide. Option 3 is the process occurs in the mitochondria of yeast cells. And 4 is it is a anaerobic process. So what do you think might be the correct answer? 1, 2, and 4. So let's see what the answer is. So yes, the answer is in fact 1, 2, and 4. It is carried out by yeast such as Saccharomyces cerevisiae. Alcohol fermentation produces ethanol and carbon dioxide. Yes. So the pyruvate that is produced in the glycolysis is converted into alcohol, right? And CO2. Right. So it is an anaerobic process. It does not require the presence of oxygen. It is a process that occurs in the mitochondria of yeast, which is not correct. It occurs in the cytoplasm of yeast, right? So only 1, 2, and 4 are correct. So I'm assuming there are no doubts in this question, right? So I'll move on to question number 13. Question number 13 is which of the following statements about industrial application of fermentation is or are correct? There are four options given to you. Fermentation is used in the production of antibiotics. Fermentation produces processes are limited to food and beverage industry. Bioethanol, a renewable energy source, is produced via fermentation, and fermentation is employed in waste treatment. So, which of these do you think is correct? Alright, so let's see what the answer is. So the correct answer is 1, 3, and 4. Not 1, 2, and 4, but 1, 3, and 4. Fermentation is used for the production of antibiotics, that is correct. So what you do is you genetically transform a bacterial cell with the genes that is required for the production of antibiotics, or you can use mammalian cells, right? So not antibiotics, but there are certain products such as insulin, etc., that can be produced by microbial cells. But antibiotics, etc., you will have mammalian cells which have the genes for the production of these antibiotics, and they use the fermentative technology of the bioreactor, etc., for the production of antibiotics, right? Bioethanol. So in case of like you're making wine using fermentation, which is a mixture of the juice and alcohol that is formed by the yeast, right? So bioethanol is also formed similarly. So you can use different kind of agricultural wastes, and using bacteria or fungus, you which can convert these wastes into ethanol. You can get bioethanol, which is a renewable energy source, right? So fermentation is employed in waste treatment, which is also correct. So if you have some dangerous chemicals in the waste, or chemicals that cannot be directly introduced into the environment, what you do is you introduce inoculum of organisms into the waste, which can convert this dangerous chemical or compound into something that can be released into the environment, right? So the only one which is incorrect is fermentation processes are limited to food and beverage industry, which is not correct. So as you can see in the question itself, it is used in waste treatment and in antibiotic production, right? So it is not limited to food and beverage industry. So that's the only thing that is incorrect. Any doubts in this question? All right. So I'll move on to question number 14, which is a match the following kind of question. And you have microorganisms in column 1 and the role in fermentation in column 2. So you have Saccharomyces cerevisiae, Lactobacillus, Acetobacter, and Clostridium. And you have in column 2 produces lactic acid from lactose, converts sugar to ethanol and CO2, oxidizes ethanol to acetic acid, and produces butyric acid. So what do you think is correct? A. So yes, let's see what the answer is. So Saccharomyces cerevisiae does convert, like we were talking about, baker's yeast converts sugar to ethanol and CO2. That's why they use in the production of wine, etc. Right? Lactobacillus, as the name itself is suggesting, is going to produce lactic acid from lactose. Acetobacter, again, as the name is indicating, will produce ethanol from will oxidize ethanol to acetic acid. That's how you get vinegar, etc. Right? And Clostridium will produce butyric acid. Right? So you are correct. So I'm assuming there are no doubts in this. Let's move on to question number 15. So question number 15 is, which of the following statements about the role of enzymes in fermentation is or are correct? So there are four options given to you. Option 1 is enzymes can act as catalysts in fermentation reaction. Option 2, enzymes are consumed during the fermentation process. Option 3, specific enzymes are responsible for the breakdown of substrates. And option 4, enzyme activity can be influenced by pH and temperature. So out of these, which do you think is or are correct? 1, 3, and 4. Let's see what the answer is. Yes. So enzymes act as a catalyst in fermentation reaction. That is correct. Specific enzymes are responsible for the big breakdown of substrate, which is also correct. Enzyme activity can be influenced by pH and temperature, which is also correct. But the only thing which was incorrect is enzymes are consumed during the fermentation process. So if you remember the definition of catalyst that we've studied in chemistry, right? So it is something that will increase the rate of the reaction without itself being consumed. So since enzymes are also catalysts, they are not consumed during the fermentation process. So option 2 is incorrect. Any doubts in this question? All right. So I'm not seeing any doubts. Just do write the doubts if you have any, right? So let's move on to question number 16. Question number 16 is, which of the following statement is or are correct regarding carbonation? So there are four options given to you. Carbonation helps to protect the aroma and color and is used to increase the taste and smell. Option 2, carbonated drinks may even enhance digestion by improving swallowing ability and reducing constipation. Option 3, under suitable conditions, CO2 has a preserving property by the inhibition of development of harmful aerobic microorganisms. And option 4, the plain sparkling water is harmful to health because of carbonation. So you have to figure out which is correct or which are correct. Do you want to try guessing what the answer is? Alright, 1, 2, and 3. So let's see what the answer is. Yes, 1, 2, and 3 is correct. Carbonation does help protect the aroma, color, and is used to increase the taste and smell of liquids. A carbonated drink may even enhance digestion by improving swallowing ability and reducing constipation. Under suitable conditions, CO2 will have preserving properties because to carbonate a drink, you have to first remove oxygen from the drink, right, or any other gases. So that since there can be only a certain amount of gas in a liquid, you have to remove that gas to introduce another form of gas, which is the CO2. So now, since oxygen is not present in the liquid as much, aerobic microorganisms which require oxygen for its respiration will not be able to grow in it. So plain sparkling water is harmful to health is incorrect. So sparkling drinks such as cola, etc., have been known to affect bone health, but sparkling water is not harmful to health because it doesn't have any added flavor. Just the carbonation itself is not dangerous, right? Any doubts in this question? All right. So let's move on to question number 17. Question number 17 is, which of the following statements about the need for de-aerators during carbonation is or are correct? Option 1 is, de-aerators are necessary for beer, cider, and wine. Option 2, de-aerators remove naturally dissolved atmospheric air from water. Option 3, without de-aeration, the water will not be saturated correctly with CO2. And option 4, de-aerators prevent the drink from going flat immediately after opening. So out of these four options, which do you think is or are correct? All right, 1, 2, and 3. So let's see what the answer is. So the correct answer is in fact 2, 3, and 4. So de-aerators are necessary for beer, cider, and wine is not correct because beer, cider, and wine are formed during the fermentation process, right? So the CO2 is formed inside the wine or the beer or cider itself. So it doesn't need to be deaerated. So after it is produced, if any deaeration is done, it will remove the, you know, air inside it, which is not good for the taste of the wine, beer, or cider, right? Um, because cider and beer are, you know, sparkling drinks. You can have sparkling wine itself, right? So deaerators will remove naturally dissolved atmospheric air from the water so that you can carbonate the drink, right? So, and without deaeration, the water will not be saturated correctly with CO2, because there's a limit to how much air can be present inside a liquid. So unless you remove the air that is already present, you cannot add another kind of air. And deaerators prevent the drink from going flat immediately after opening is also correct, right? So any doubts in this question? All right, so let's move on to question number 18. You have to match the type of acid given in column 1 with its principal use in soft drink in column 2. So you have citric acid, phosphoric acid, tartaric acid, and malic acid. And in column 2, you have gives different flavor compared to citric acid, used in colas, used in apple flavored drinks, and commonly used in lemon flavored drinks. So if it is about flavor, you have to think of which acid enhances the taste of that particular drink, right? So do you want to try guessing the answer? Alright, I'm not seeing any answer, so let's maybe see what it is. I think it is not working properly, so let's see in B. Alright. So citric acid is in fact commonly used in lemon flavored drinks because the citric acid is what gives it the distinct taste, right? B is phosphoric acid, which is used in colas. Tartaric acid is used as a, you know, different flavor compared to citric acid, and malic acid is used in apple flavored drinks. So your correct answer B is correct, right? So the next slide was not working. So I just answered in this. So you both are correct. It is answer B itself. So maybe let's move on to question number 19.